Hello, my name is Christina Smith, and this week I will be discussing Article 7 presentation, which is the chemical modulation of transcriptionally enriched signaling pathways to optimize the conversion of fibroblasts into neurons. And this was a collaborative study across the Laboratory of Genetics in La Jolla, California, the University Hospital Freiburg in Germany, the Liber Institute for Brain Development in Baltimore, and the Institute of Molecular Biology in Austria. So I want to discuss the overall overview of the presentation. First, I will introduce the background of the comparison between cellular reprogramming versus direct reprogramming. This is due to the fact that I want to bring up and highlight important differences between these two methods and why scientists within this article decided to use direct reprogramming instead of cellular reprogramming. Then I want to discuss the purpose of the study, because there are multiple points that the authors address and test. Then I want to head into a detailed discussion of the various figures in this article. I will be discussing Figure 1, Figure 2, Supplementary Figure 1 for Figure 2, Figure 3, and then Figure 5. After a detailed discussion, I want to talk with you guys about my proposed additional experiment to further along the author's hypothesis and intended future work. Afterwards, I will conclude the presentation with a summary of the article and future works that the authors propose to do. To start off with, I want to discuss the background of cellular reprogramming. We discussed this method in various modules so far in class, but I wanted to give you a brief rundown on what it is and to give you a refresher. If you look over to the figure on the left-hand side, it shows a depiction of normal development versus cellular reprogramming. So typically, in normal development, which is depicted by figure A, it is often compared to a ball rolling down a hill. This is a cell going through a developmental landscape from an undifferentiated stem or progenitor cell state to a physiological mature final cell state. And this is how all cells in the body begin. You start off with a few cells that form a zygote and then to an embryo, after which the embryo begins to cluster cells together that have the same function to create tissues. And then tissues become organs that have one final function that work together. Cellular reprogramming, on the other hand, flips this pathway backwards. So in Figure B, it shows a process whereby cells travel back up their maturation pathway through the epigenetic landscape to become more immature cells or pluripotent cells. And this happens through numerous intermediate steps and needs chemicals and specific transcription factors to do so. Next, I want to discuss direct cell reprogramming. This type of reprogram is unique due to allowing the patient's cell to jump directly to a specialized multipotent stem cell of choice from the terminally differentiated cell. As I said in the previous slide, cellular reprogramming requires multiple intermediate steps to achieve the induced pluripotent stem cell fate. So instead of making pluripotent stem cells from patient cells and differentiating them to multipotent stem cells over an extended period of time, direct cell reprogramming produces directly the specialized multipotent stem cell of choice. This typically costs less than cellular reprogramming because scientists do not need to buy or create different transcriptional factors to push the cell back into the pluripotent state. In addition, it is safer and less time-consuming than cellular reprogramming. For these particular reasons, scientists in this study decided to use direct cell reprogramming in order to see how fibroblasts convert back into the induced neurons. So these figures below depict more on direct cell reprogramming. The figure on the left-hand side typically depicts cellular reprogramming. So as I said before, cellular reprogramming can also be called rejuvenation. So it's taking a differentiated stem cell, moving it to an intermediate progenitor state to the pluripotent state. And once scientists have reached this pluripotent state, they can then manipulate that cell back into a different differentiated cell. So it needs to move back into an intermediate progenitor state to another state where they can then differentiate that cell into the specific cell they want. And so, as you could tell, this is a very long process. In addition, direct cell reprogramming can also achieve multiple cell state types. So the figure on the right hand side shows various cell lines that can be directly programmed into different cells. So B cells can be turned into macrophages, muscle precursors can be turned into brown fat cells, fibroblasts can be turned into cardiomyocytes. However, within this particular article, we are looking at fibroblasts being turned into neurons. And as you see along the arrow, there are different transcription factors that are required in order to convert these cells into the differentiated stem cell of choice. And so I wanted to discuss the importance of direct reprogramming in this article to give you a summary of it. So by overexpressing pro-neuronal transcription factors, human somatic cells can be converted into the induced neurons, and which are typically INs. So the advantages of this method, as I said before, it can bypass a pluripotent stage, but it can also bypass any other stem cell-like stages and preserve the epigenetic information of their donor age. And this is very, very important. By preserving the epigenetic information of their donor's age, scientists can use this to study age-related disorders. In addition to this, there has been recent research that induced neurons can also replace damaged cells following brain injury. Although this is a rough, rudimentary hypothesis right now, and it's still being tested, scientists have found that this could be used to help these individuals that are brain damaged. So now I want to discuss the purpose of this study. There are four main purposes that I found while reading this article. The first is to increase the efficiency of direct reprogramming by combining two transcriptional factors, NGN2 and ASCL1, in the same vector. The second one is identifying molecular pathways that are upregulated and what pathways are activated during reprogramming. The third is to determine whether or not transcription factors differ across fibroblast cell lines and what happens when we add them versus when we don't. And the last is to determine whether or not direct reprogramming can retain epigenetic age of pluripotent stem cells. As I've said in the previous slide, retaining this epigenetic age is important in order to study age-related disorders. So I want to give you an introduction on NGN2 and ACSL1. These are very important factors in converting fibroblasts into induced neurons. So NGN2 is a BHLH transcription factor involved in both neurogenesis and neural specification. So what is ASCL1? This protein plays a role in the neuronal commitment and differentiation and in the generation of olfactory and autonomic neurons. So in previous studies, not the one that I'm discussing right now, commonly NGN2 and ASCL1 were not efficiently delivered into fibroblasts due to their construct. In addition to this, they have a robust transgene expression. This all leads to the likelihood that these two transcription molecules are not actually introduced into fibroblasts, and therefore they cannot be converted into induced neurons. So as I said before, typically NGN2 and ACSL1 are distributed across several lentiviral vectors. However, given that the lentiviral transduction is not 100% efficient, and because an individual fibroblast usually needs 2 or more viruses to survive chemical selection and successful reprogramming, previous studies have left room to improve this vector construct. So, to eliminate the need for co-infection, scientists within this particular article combined multiple cassettes to yield the UNA construct, which is depicted in Figure A. This construct adds both NGN2 and ASCL1 in the same vector in hopes that will increase the efficiency of converting fibroblasts into induced neurons. And just as a side note, both NGN2 and ACSL1 proteins to be transcribed, specific promoters need to be included in the vector for both proteins to be made. So now I want to move on to figure 1b, c, and d. So in figure 1b, this typically shows cell lines that have high, medium, and low performance in directory programming. So fibroblast cell lines that can convert a lot of fibroblasts into induced neurons are indicated in green, medium performance are indicated in blue, and fibroblast cell lines that do not convert fibroblasts into induced neurons or at a very low level are indicated in red. Figure 1c indicates immunocytochemical analysis of the induced neurons following 3 weeks of conversions stained with beta-3 tubulin, PSA-NCAM, and DAPI. And this just shows a visual representation of the neuronal markers that we will discuss further on. So in figure 1d, flow cytometry actually revealed that UNA-derived induced neurons exhibited significantly more PSA-CAM positive cells than the previous construct, which is depicted by E plus 2NA. So this can actually boost the efficiency by up to 90 to 100% for 2 suboptimal fibroblast lines, which basically means those of low performance in converting fibroblasts into induced neurons. So this is very exciting. But it can also increase the efficiency of lines that already converted well by 30 to 80%. So the fact that it can convert cells that already have high performance more is very, very good for this research. Now I want to move on to figure 1e and 1f. Figure 1e describes a comparison on PSA and CAM positive cells in the new construct UNA versus the old construct. These cell lines chosen in this particular experiment are L2, L5, and L7. And if you remember from figure 1b, these cell lines depict high, medium, and low performance respectively. Scientists wanted to show that no matter the performance level of fibroblast cell lines, the new construct UNA can increase the efficiency of directory programming. And as you can see, low-performing cell lines increased efficiency by expressing PSA and CAM over 100%. And even with high-performing cell lines, there is still an increased efficiency by 16%. This shows that scientists were able to successfully demonstrate their construct to increase directory programming. Moving on to figure 1f. This describes the immunocytochemical co-staining for the neuronal markers beta-3 tubulin and NEUN. The top row shows the co-staining in the old construct, while the bottom row shows the co-staining in the new UNA construct. Visually, you can see a higher degree of expression in the new construct for both neuronal markers. And in fact, scientists were able to show that the new UNA-induced neuron construct were on average twice as likely to be positive for NEUN and beta-3 tubulin. This is just another factor that shows the increased efficiency of directory programming by combining NGN2 and ASCL1 transcription factors in the same vector. Lastly, I want to discuss figure 1G and 1H for the first experiment. So these figures show the quantification of the immunocytochemical staining. Figure 1G shows again how different cell lines react to the new UNA vector when staining for the neuronal markers beta-3 and NEN. Figure 1G shows the quantification for beta-3 tubulin neuronal markers for cell lines L2, L5, and L7. And again, remember, these lines correspond to high performance, medium performance, and low performance of fibroblast lines. Interestingly, the high performance fibroblast line L2 increased more than previously shown by 37%. It is still dramatic for low performance cell lines L7 by increasing efficiency by 93%. Figure 1H shows the quantification of the immunocytochemical staining using DAPI. Again, the low performance cell line increased dramatically over 177%. Altogether, experiments from Figure 1 showed the use of a single all-in-one lentiviral vector UNA, and it was easier and less prone to experimental error.